Great pleasure to announce our first keynote speaker, Reinhard Jahn from the Max Planck Institute for Biophysical Chemistry in Göttingen, Germany. And he will talk to us tonight about exocytosis in neurons and looking at the interplay between proteins and lipids. Welcome very much, Reinhard.
Okay, all right. So first of all, I would like to thank the organizers for inviting me to this very exciting meeting. I should say this is not often that I give a talk on the day when I'm arriving from international flights, so I hope that things are going reasonably well. If I start to talk nonsense, we should make unpleasant noises so that I start noticing it. Anyway, so what I would like to talk to you about is there's something. Second here is still. All right. Is giving you first a brief overview over our field of interest, and this essentially relates to exocytosis in neurons. And for those of you who are not neuroscientists, to bring you up to speed, a typical nerve cell, this is here a motor neuron, has a particular morphology. So it has a kind of soma or cell body region with a process called dendrites, so-called somatodendritic regions, which also serves is the antenna region of a neuron. So this is where the signals are coming in from many other neurons. They're integrated, and then here at this point, the axon hillock, they are converted into fairly primitive binary codes, the so-called action potentials, which then travel down the axon, and then once they reach the end of the axon, they are reconverted into a chemical signal, and this is what I would like to focus upon. Because what happens at the synapse here is that when a presynaptic action potential arrives, the presynaptic membrane depolarizes, and then the signal molecule we have heard about in the previous talk, calcium, comes in through voltage-gated calcium channels with a very short delay time, which can be as short as 50 to 100 microseconds. Calcium triggers the exocytotic fusion of synaptic vesicles with the presynaptic plasma membrane. These synaptic vesicles are the storage organelle of the neurotransmitters, and once they are released, they diffuse across synaptic cleft, bind to postsynaptic receptors, and then activate these receptors by changing the electrical properties, either by activating ion channels or else by activating also second messenger cascades. So what I'm interested in is this particular reaction. So this little piece here, this one arrow, is the one which I would like to focus on a little bit more. Now, the basis of exocytosis is a process called membrane fusion. On membrane fusion, the task the cell has to manage is to convert a stage from here. We have two separate bilayers from the vesicle and the plasma membrane in this case to end up here, when there is an aqueous connection between the vesicle lumen and the outside of the cell, which is surrounded by membranes. And the question is now how to get there, and second is how are the proteins actually mediating this process. And I should say before, and I will come back to this, that we have really not much of information about this. So we really don't know what the energy barrier is. There are quite a few numbers running around the literature, but none of them is experimentally verified. So most of these numbers are model dependent. So we don't really know how much energy is required to fuse a membrane. And the second is that we don't really know about the intermediate states, in particular what the energy barriers are for an intermediate stage and what the intermediates are where there might be, for instance, a status where the membrane can be arrested at intermediate state in the fusion reaction. Now, the proteins we know now in neurons that carry out the jobs are the so-called synaptic snare proteins. Synaptic snare proteins are small transmembrane proteins. So there are three of them called syntaxin, synaptobrevin or VAMP, and SNAP25. These proteins have funny names because the names were given to them before anyone had any idea what these proteins were doing. And so at that time, we're talking, early mid-80s, that once you had a new protein, you clone it, you own it, you gave the fancy name. And I can say so without offending anyone because I'm not innocent of one of these names. Anyway, so these three proteins are highly conserved in evolution. We talk about 80% sequence identity between C. elegans and mammals. They're the founding members of the Snare superfamily of proteins, which we now know operates in most, but not all, intercellular fusion reactions in eukaryotes. They interact with more than a dozen other proteins known to be involved in exocytosis. And they are cleaved by tetanus botulinum neurotoxins. These are powerful bacterial toxins so-called AB toxins, in which the light chain is a protease with a very specific taste. 5 of these toxins cleave synaptobrevin, 2 cleave SNAP25, and 1 cleaves syntaxin. So this is what I also call the poor man's knockout. So we have a very nice toolkit for studying these synaptic snares by using the light chains of these toxins. Now, work in the last years, prominently started by Jim Rothman, now at Yale, has shown that these snares undergo an assembly-disassembly cycle that is intimately related to the fusion reaction. So we have, as you might remember, 1 snare on the synaptic vesicle, 2 snares in the plasma membrane. These snares form acceptor complexes, which then interact with the N-terminal end of the synaptobrevin in the synaptic vesicles. In this way, these proteins gradually assemble. This is a spontaneous, highly exergonic reaction in which a lot of energy is being released. And this kind of pulling together, this is a zippering reaction, as you will see a little later in more detail, then forces the membranes towards each other. And this, as we think, and many other people also think now, is the main reaction overcoming the energy barrier for the fusion reaction. Then fusion is occurring, so the fusion pore opens, the snares convert in from the so-called trans to the cis conformation. They have spent their energy, so are now in a relaxed state. They need to be re-energized, and this is done by a complex consisting of a protein called alpha SNAP and a AAA ATPase NSF. AAA ATPases very often have a job to disentangle protein aggregates, so this is quite up to style that AAA ATPase here is using its energy to rip apart the snare complex, thus re-energizing them for another round of exo-endocytotic membrane fusion. Now, the crystal structure of the snare complex nicely confirms the zippering model because you see that there are elongated helical bundles. We have shown years ago that the reaction is indeed initiated at the N-terminal end of this reaction, and I should say that when we first published this structure together with Axel Brunger, the structure ended here, so we drew this in the cartoon. We got a lot of beating because everyone said, well, you don't know whether these are helical structures. And there was a 10-year long debate until I found a very talented graduate student named Alexander Stein who finally managed to crystallize the full-length snare complex. And to our, I should say, to our fun, we really realized this is a contiguous helix extending into the membrane, just very nicely confirming the zippering concept of snare function. Now, my lab in the last years is interested in understanding the details of the reaction, to understand what are the intermediates, how is the reaction regulated. And the approach we are taking as biochemists that we essentially use artificial membranes. We also use native membranes, but in most cases we use artificial membranes in which we reconstitute fusion ab initio by putting in snare proteins or other proteins and then monitor fusion with a variety of different assays, which I don't want to go into too much detail. Most of them are fluorescent based. Now, the first publication using this type of assay was again from Jim Rothman's lab. You see here from his original paper, he saw a very nice fusion reaction when the complementary sets of snares were present in these membranes. But the problem was the time scale, because it took about almost 5 minutes before the reaction even started. And then it was a very, very slow reaction. And this was kind of a problem for the field because these are the snares that are supposed to mediate sub-millisecond rate exocytosis in neurons. Now, work of the years in my lab and in other labs have shown that these snares, in particular the ones on the plasma membrane, undergo a very complicated association-dissociation equilibria. For each of these states, which I don't want to go into too much detail, there is experimental evidence. And I would like to point out that in particular the protein syntaxin forms clusters in the membrane which are stabilized by PIP2, as you have heard in the previous talk, is an organizing lipid in this case. And, but there is an equilibrium between clustered and free syntaxin, and also the association between syntaxin and SNAP25 can take several forms. And it's pretty evident that only this one is an active acceptor complex, and this one seems to be highly regulated by an array of additional proteins, but we don't understand the mechanism yet. So what we have tried to do is to look at the later stages of the reaction, is to simplify this. This is work by Dirk Fassauer, a group leader, former group leader in my department, who came up with a trick. He said, well, why don't we build a snare complex in which the C-terminal portion of the complex is occupied with a fragment of synaptobrevin. Now, this would leave the N-terminal part free for the synaptobrevin to nucleate. And the original idea was just to measure the binding kinetics. But there we were a bit lucky because it turned out that the protein doesn't only bind with very fast kinetics. We need stop flow to measure this. But also it's capable of kicking out this peptide this way completing the zippering reaction and actually mediating membrane fusion. So we get fast fusion, but the price we obviously pay is that we have to overcome the displacement energy of this competitive inhibitor in the complex, and keep that in mind because this is becoming important. You see that if you use this complex, you get a much faster fusion reaction, at least 2 orders of magnitude faster. So we have successfully bypassed these upstream control reactions by simply creating a kind of homogeneous type of exactor complex which can then be utilized in the fusion reaction. Now the question is now, how do they do the job? What is exactly going on during the fusion reaction when the snares do this? And you might say, well, I mean, they are pulling here, and this is something which for quite a few years I was actually also believing by myself. And let me just get back to this, but before I go into this, let me discuss with you briefly one of the kind of core questions. How many complexes do you need to fuse the membrane? Well, this was open, and when we started out a few years ago, you see that the literature numbers ranging from 3 to 15 were published. So there was really not clear evidence how many snares would be required. Now, what we did, and this is a work of Geert van den Boogaard when he was in the lab, he simply made an experiment in which he diluted the snares more and more in the membrane, and used fluorescently labeled snares in which we had stoichiometric labeling on the snare proteins, which is very important, obviously, for that experiment to make sure that you have stoichiometry with the fluorescence tag. And this way, once you dilute further, you can actually then do single molecule photo bleaching to count the molecules on the vesicles by simply plating them out on a cover slip and just looking at the bleaching steps. And this way, he was able to show that the number of snares more or less corresponded to what we had calculated. It was actually a little lower. And also, he could show that the distribution is perfect. It corresponds to perfect Poisson distribution, which means that at least at this low concentration, we don't have aggregates or oligomers. They just behave as single molecular entities. And he was able to create at its highest dilution, 1 to 16,000, this is the ratio between snares and phospholipids, a situation where a majority of liposomes had no snare. And since they were not labeled, we didn't really care much about them. But there was the majority actually of the ones having snares had only a single snare complex. And to make a long story short, all of them were capable of fusing. So it turned out that the single snare complex is sufficient to mediate fusion of small liposomes. Now this is, we did a lot of controls to convince ourselves that we're not looking at artifacts. So we see the same data when we use purified synaptic vesicles from the brain instead of liposomes as fusion partners. So it works at least with one biological members on one side. And we also confirmed that contact mixing assays show that full fusion occurs with a single snare complex as well. So we not only have lipid mixing, but also content mixing for the experts. This is quite important because it means that any kind of mechanistic model which requires any type of symmetry in the snare organization around the fusion pore cannot be correct. That's, I think, one of the most important conclusions, even though I should hasten to add that we know now that when we use larger liposomes, we need more snares to overcome the barrier. So since we published this work a little later, there was 2 confirmations in biological systems, in particular this work, in which labeled synaptobrevin was reconstituted in neurons from knockout strains which didn't have synaptobrevin, showed that in this case you need 2 synaptobrevin molecules, which is close enough. In this case, obviously, we have all the regulatory proteins siphoning off some energy, so that makes a lot of sense. Now, it makes sense not only because it's fitting the experiments, but it also makes sense biologically because in neuroscience, we are sometimes, first of all, I should point out that the snare complexes operating in neuronal exocytosis and the ones operating in other fusion events inside the cells look perfectly identical. I mean, the structures are really getting boring. We saw 3 of them. Axel then did the 4th one, and they all look the same. So these are all 4 helical bundles. There's very similar positions of the individual chains and so on. Stability, this is a little bit less stable, but these are also of a similar stability. So even the energy contribution during assembly seems to be the same. So there is not much in terms of what is different. What is different, however, is the abundance of these snares in the different fusion reactions. So in neurons, you have on the synaptic vesicle, this is a model of a synaptic vesicle, we have 70 copies of synaptobrevin. So it's the most abundant synaptic vesicle protein. You see this here modeled in a kind of conformational ensemble. And also in the plasma membrane, the concentration of SNAP25 and syntaxin is extremely high. So we have here clusters of 70 to 90 molecules of syntaxin in the plasma membrane. So here we have a lot of these snares sitting. So there is, whenever a vesicle gets closed, there's always at least several snares within interaction distance without the need for any kind of sequestering or accumulating it on one side. Now, in endosomes, however, in the synaptic vesicles recycle through endosomes, and we have studied these endosomal snares quite a bit. These endosomes fuse. They fuse in a snare-mediated way. The complex was on one of the previous slides. Even though the neuronal, the exototic snares recycle, they are not involved in this fusion reaction. So you can use an endosomal fusion assay. You can treat them with certain neurotoxins to shave off all the neuronal snares. They still fuse happily using their endosomal snares, which we know. These are VAM4, VTI1A, syntaxin 6, and so on. And there are only a few copies on the endosomes. So we need only, in particular for VAM4, we have only 4, 5, 6 copies on the entire endosome. That means this fusion reaction must be efficient with only very few snares, 1 or 2. So that makes biological sense. Now back to the questions, how do they do the job? How do they fuse membranes? Now again, coming back, the original idea, which I subscribed to for quite some time, is that this is kind of a nanomechanical device. A nanomechanical device which creates tension. This tension is then transmitted to the contact site, destabilizes the membrane, and then overcomes the energy barrier this way. Now, before going into this, let me just give you a little bit of a history of how membrane fusion was seen. And this is sometimes, when you get older like myself, sometimes interesting to look at back in the old papers. And I mean, this is one of the first papers I could trace from George Palladie, who really has studied this were actually, interestingly, endothelial cells, where used conventional electron microscopy to really look at intermediates, which we would nowadays interpret as tightly docked, hemifused, and this was a situation where you found always some material between the vesicle and the outside world, which you couldn't really quite interpret. I mean, you also see that these were different times, because this was a paper in Journal of Cell Biology, figure number 28, and try to publish a paper with 28 figures in Journal of Cell Biology nowadays. I think you will have not much of a success. Anyway, now in the late 70s, people were realizing, well, membranes are bilayers, and there were actually some interesting theories. I just got this from an old volume, which I found in my personal library on the shelf from Scientific American from 1979. I think Misha might remember this. And where, at this point, the idea was that there must have been some isotropic micellar phases, intermediates. And these were kind of ideas how fusion might work. And you already see the problems. This cannot be stable. This must be leaky. And also, there was evidence that lipids or amphiphils promoting micellar phases are actually among the strongest inhibitors of in vitro fusion reactions. So you can use, lysophosphatids, which would promote micellar phase, and they are very efficient inhibitors of any type of in vitro fusion reaction. Now, so the model which then was actually put forward, and I'm really glad to say that Misha Kozlov, who was one of the initiators of the model, sitting in front of me, and you will hear him later, talking about membrane properties in the endoplasmic reticulum. And this model then describes the fusion reaction as an ordered sequence of steps, where first the proximal monolayers fuse with each other, then we create this kind of symmetrical intermediate, which you might also refer as a kind of a stalk intermediate. And this stalk intermediate for this is quite a bit of experimental evidence now from both NMR and low-angle X-ray scattering. And then finally, this stalk breaks open either via an intermediate hemifusion diaphragm or directly by simply connecting these membranes in trans and break them open. Now, this is the model which, by and large, is being accepted today, even though the energetic, let's say, pathway of the reaction is still, I think, somewhat under debate. And I should mention that nowadays, new techniques such as molecular dynamic simulations, coarse-grained simulations, have actually given us another physical access to modeling membrane fusion, but I would say that on the first glance, the intermediates are compatible with the stalk model, even though they are a little bit more chaotic, which one would expect from these type of molecular models. Again, the question is, where are the energy barriers? Where are the energy minima? Are there any energy minima along the fusion pathway? Now, the elastic theory does more than just coming up with a nice picture, because the elastic theory describes membrane as bendable sheets. Bendable sheets means that, like, if you have a membrane which there is a spontaneous curvature in the membrane, if you start bending it, you can use fairly standard equations to describe the energy which is required. And this is kind of equation which is important because it describes the force which is required to bend the membrane as a derivative of the actual curvature, which needs to be calculated, which is not simple, and spontaneous curvature. This is important to keep in mind that every membrane has a spontaneous curvature. Artificial membranes, the spontaneous curvature of a flat membrane is always 0 because it's symmetrical, but biological membranes are not symmetrical, so the spontaneous curvature is very likely to be different. And the second assumption, which I think is something which one needs to be discussed, is that the bending modulus, kappa here, is considered to be constant over the plane of the membrane, but very clearly at these contact sites, this rule doesn't apply anymore. And I don't want to go any further into this. Anyway, again, taking from a nice paper by Leonid Chernomyrdin and Michel, how could proteins overcome the energy barriers? There were several models. I don't want to discuss them in detail, but one of them, which looks particularly appealing to a Snare person, is this one, where we have proteins sticking in, extending into the membrane of both membranes, and then basically creating some kind of a mechanical force. And if you look at this, let me just mention that one of the problems we have is that we don't know the spontaneous curvature of synaptic vesicle. You see the highly asymmetric membrane composition. So it's very likely that the spontaneous curvature is positive. And I would really love to measure this, to be honest. I mean, I'm an experimentalist. I would love to measure the spontaneous curvature of synaptic vesicle, but so far I don't have any way of doing this. I mean, I talked to lots of physicists and so on. It's not a trivial problem at this point. So the snares are actually quite well set up, as discussed, to follow this type of model. And you see here intermediate reaction. They are sitting between the membranes. They might actually guide the membrane through these kind of intermediates as predicted here. So they would be perfectly well suited to be force generated. But the question is, do they generate force at this partially fully zippered state? Do they still, extend stress on the membrane? Now, to address this question, we had a little bit of luck because we started to use larger liposomes where fusion is a bit slower. As I mentioned, you need a little bit more snares. We also diluted the snares in the membrane to a much lower value. So we use usually now a snare ratio, phospholipid to snare ratio of 1,000 to 1. Keep in mind that synaptic vesicles have a snare ratio, of 100 to 1. So there's 10 times more snares in synaptic vesicles. But this allows us to kind of slow down the reaction a little bit. And it turns out, if you look, you get very nice fusion reaction. But you see here, if you magnify this, you see that at the onset of the fusion, there's a delay. There's a delay phase here. So we took a closer look at this delay phase. And this is the work of a very talented student named Matias Hernandez. And it turned out this delay phase is due to the fact that the snares start nucleating, but there is a kind of delay which we very likely is due to the fact that we need to kick out this inhibitory peptide in our fusion reaction. Now, that was confirmed when we looked at N-terminal association. So we had used various fluorescence assays to show that the N-terminal association occurs without a delay between the snares. So here we don't have this delay, only in the fusion reaction. Now, if you then look, distinguish between fusion and docking, and this is a single particle, this is fluorescence cross-correlation spectroscopy, single particle assay, you see there's you see an accumulation of a docked vesicle intermediate. So this is total docking, this is the calculated docked intermediate, and this is fusion, and you see that these docked intermediates is then consumed in the course of the reaction. And we also looked for fusion and hemifusion. This is only for the experts in the audience, so we also found a certain degree of hemifusion. We consider hemifusion, extended hemifusion, as a dead-end reaction, which is actually quite a nice agreement with studies done many years ago by Leonid Chernomyrdin on viral fusion proteins. So also a dead-end intermediate hemifusion state was identified. Now, we then did cryo-electron microscopy in the States, and there we were actually quite surprised. So we identified in this particular peak here, we identified a lot of profiles like this, and also profiles like this. These are hemifusion profiles. This is cryo-M, no fixation, just, shock frozen. And so we are fairly confident that it represents the state of the vesicles in our solution. Now, this is a bit surprising, because this is an intermediate, and if you look at this, it has a very, very high degree of curvature here. I mean, we calculated the curvature radius was about, less than 10 nanometers, probably 5 nanometers, which would be extremely unstable for this membrane. As yet, these vesicles, as I will show you in a little while, can be stable for 10 minutes. We can actually stop the reaction at this point, and then we have these intermediates in the cryo-M sitting there for a long time. And this is very likely due to the fact that we have lipid mixtures, we have lateral diffusion of lipids. Lipids will kind of soften this kind of kink and allowing it to be more stable. Now, what we think, because this is an on-pathway intermediate, these intermediates are consumed during the course of the reaction. What we think is going on here is that what the snares do, They bring these membranes that close to each other, and that's their main job. They need to bring the membranes close. Then once they are very close, you overcome the hydration barrier, but then the next step would be basically the breaking this hydration barrier. There may be a phospholipid molecule or two jumping over with the phospholipid tails. This would be the activation energy barrier, and then it would go downhill to the stalk and then finally to this fusion reaction. Now, so we think that close proximity and the associated dehydration of the membrane contact zone constitutes the main energy barrier, as already suggested, by the way, many, many years ago. And for the snares, it means that the linker stiffness may not be that critical, and rather local lipid perturbation after zippering. So what the snares need to do is to bring the membranes close, and that also explains why the linker and also transmembrane domains in snares tolerate quite a bit of modification. So you can metabolize them. They will eventually affect the functionality, but they're not that sensitive, not nearly as sensitive as the zippering part to mutations, which might even disrupt helical structure and so on. Okay, so we don't think they act, at least in the late stage, act like a spring. Rather, we think that they are, this is now taken from coarse grain simulation done by colleagues of mine in Göttingen, that they act by bringing the membranes very close to each other, and then essentially the jumping over here at this point, here we get this kind of stalk intermediate which proceeds in the simulation to the fusion reaction. I would like to mention one more point, and that relates to the fact that the transmembrane domains of the snares are a little bit too short for the plasma membrane. We didn't notice this first. Actually, the first hint we got was from when we tried to model the crystallized complex into membrane, and you see here these deficiencies here, because the crystal structure, at least for syntaxine, includes the entire transmembrane domain with the only exception of the very last amino acid. And even if you model that in, no matter how you move the protein, it's a little bit too short. So we now have actually following this up, and we realize that there is a kind of a small hydrophobic defect, and it is possible, even though we are not sure yet, that this small hydrophobic defect might also contribute to the fusiogenicity of the snares at this contact site. Okay, I think I have 10 more minutes, and I would like to use these minutes to switch to the question of calcium. We had talked about calcium in the previous talk. I told you about calcium. So far, I told you all about the snares, but after all, in neurons, the snares are regulated by calcium. Now, the protein which is doing this is a protein called Synaptotagmin. Synaptotagmin was originally identified in a collaborative work between Tom Sudov and myself many, many years ago, and we then showed that it's a calcium-binding protein. At that point, it contained so-called C2 domains, and at that point, the only other protein known to have C2 domains was protein kinase C. And the protein kinase C, it was known that these C2 domains bind calcium. We were very excited. We showed that this is also binding calcium. We then hypothesized that this might be the calcium sensor. Now, to cut a long story short, this turned out to be correct. So the protein has been knocked out in every conceivable species. The phenotype is always the same, that you lose the fast phase of neurotransmitter release. So the calcium-dependent fast phase of neurotransmitter release is completely dependent on synaptotagmin. Now, these two C2 domains bind each 3 calcium ions. Calcium triggers binding to acidic phospholipids. So this is one thing which has been shown, and we have actually shown that it particularly well likes to bind to PEP2, the phospholipid which was already mentioned. But also it binds to individual snares, to Q snares, to binary and ternary snare complexes in the absence and presence of calcium, which makes the molecular understanding of this molecule actually quite complicated. Now, deletion, as mentioned, of synaptotagmin abolishes fast neurotransmitter release, and disruption of calcium binding to the C2b domain abolishes synaptotagmin function. The disruption of the other binding site is not quite as severe, and there is a separate binding site for PEP2, which is adjacent to the calcium binding site, and that also causes weakening of the calcium triggering. Now, the job and the goal is to understand how exactly does a protein operate. Now, over the years, quite a few labs have published that synaptotagmin inclusion into a snare-mediated liposome can accelerate the fusion reaction. Whenever you had a kind of a weakish activation, this was considered so important to end up in top journals. But even though one should say under physiological conditions we get an activation which is more than 100,000-fold, whereas in these reactions the best was 2, 3, perhaps 5-fold activation. So evidently something was missing there. And to cut a long story short, we don't really know yet what is missing, but at least I would like to give you some ideas. Now, one of the ideas which was put forward is that actually synaptotagmin might affect the membrane curvature. So it might go into the membrane and create some kind of a membrane bending, which would then essentially destabilize the membrane and then drive fusion forwards. So the theory are split between theories acting on the membrane and on the snares, and this is one of the membrane interaction theories. Now we thought we have this kind of nice model system with a large liposomes, where we have a retarded fusion reaction at a relatively late state. And we asked the question, is Synaptotagmin, if it kind of changes the membrane energy, we should be able to see an effect here. So what we took advantage of is a mutation in Synaptotagmin. It was an accidental finding of a single amino acid deletion. If we use this deletion, if we use small liposomes, the fusion is retarded. If we use larger liposomes, the fusion is completely blocked. And what we see are essentially an ensemble of completely docked vesicles. And if you look at them in the electron microscope, you see again this kind of intermediate where the membranes are very tightly opposed. Now the snares are fully zippered. There's no peptide here, but they cannot go further. And what we wind up is an arrested fusion reaction at this very late stage. So I love this Mickey Mouse picture here, by the way. So we have created a state in which the snares are fully zippered, the membranes are tightly docked, but in the large liposomes, they're just a little bit too weak to bring the reaction to completion. So fusion is only observed when using small, but not when using large liposomes. Now the first experiments we did is we checked whether increasing the concentration of snares helps. The answer is no. So if you increase the concentration of snares, you don't get any addition. The second question we asked is, what is Synaptotagmin doing here? And when we add Synaptotagmin, and we have done it in all conceivable ways, putting it in the transmembrane domain, in situ domains, whatever, PIP2 in one of the acceptor membranes, absolutely no effect whatsoever on the fusion reaction. So I don't think, I mean, even though I might argue with Misha, but I don't think that Synaptotagmin's effect on fusion has anything to do with changing membrane curvature at this point. So how does it work? Now, we had a kind of accidental, first starting with a fairly artificial experiment which might give us some clues. What we did here is and this is again the work of Gerd van den Bogart, a very talented postdoc in the lab. We used liposomes in a normal snare-mediated fusion reaction where we basically lowered, or having acidic phospholipids in both membranes, and then we lowered the salt concentration. What you then get is that fusion is, as lower you go, fusion comes to a complete stop. That's very simple to explain because this is charge repulsion. If you don't have enough, ions around, the liposomes repel each other, and they cannot come close enough for snares to function. And we have done all the controls to show that this is indeed the case. So you can probe for snare reactivity on the membranes by using soluble snares. They're fully reactive. If you replace these lipids with non-charged phospholipids like phosphatidylorthanolamine, phosphatidylcholine, we get nice fusion reactions. This is really charge repulsion. So a system in which liposomes simply don't get close enough. Now, when we add Synaptotagmin, the story becomes interesting. Because first of all, these liposomes, if you have PIP2, the target liposomes, these liposomes cluster, but they don't fuse. And if you then add calcium, we get a full-scale activation of the maximum fusion kinetics mediated by the snares. So what's going on here? Now, first of all, this is a situation. Unfortunately, we don't have yet cryo for technical reasons. We hope to get it, how it looks like. So I can show you only this ugly picture, negative stain picture of these clustered liposomes. So we have created a state here that simulates synaptic excitosis. Vesicles are docked, no fusion in the absence of calcium, but snares are active and massive acceleration of fusion by calcium binding to Synaptotagmin. Now the idea was that perhaps what's going on is that the membranes are still a little bit, despite Synaptotagmin linking them, are still too far apart for the snares to see each other. And what Synaptotagmin might be doing is cross-linking the membrane, just pulling them a little bit closer to each other. And I have some ideas, some support for this view. First of all, we did FRET experiments in which we labeled the lipids of the membranes with different dyes. And you see here when you now add calcium that you get an increase of FRET between the membranes, which is at least an indication of the shortening of the distance. Furthermore, we have done, so this is the idea. So we have first a trans contact made by Synaptotagmin 2, as we now know, syntaxin PIP2 clusters, which then kind of gets the vesicles attached to these sites. And then when calcium comes in, then we get cross-linking of the membranes, and then the snares become into action distance and start zippering. According to this model, the snares would be single-shot devices, so they wouldn't be partially zippered, as most people assume now, before the fusion, before calcium comes in. They wouldn't see each other before calcium enters the cell. Now, a final detail relates to the PIP2, to the different binding sites, which I was explaining to you. We have the PIP2 binding site, we have the C2B site, we have the C2A site. And we also we wanted to know, because interestingly, if you do away with this side, the C2A side, the protein is still functional. And experiments show if you just use the C2B domain, the C2B domain can cluster liposomes. The question is, how can it do this, having only one calcium binding site and a closely adjacent PEP2 binding site? At that time, let me just skip over this. Let me show you here. We have done experiments. For instance, here we used membrane sheets with syntaxin PEP2 clusters. These are native membranes. You add syntaxin. You can add either this full C2AB domain, or you also use only the C2B domain. Then you add liposomes from the outside. And you see that the liposomes are recruited by Synaptotagmin to these clustering sites, which is direct evidence that they can do the job. They can bind, they can recruit liposomes to these sites. And to reconcile this, we have recently solved the crystal structure of the C2B domain in complex with phosphoserine. Now, there was a structure already out there from protein kinase C, and we modeled this structure, and it didn't quite fit. It didn't really make much sense. We couldn't really get a kind of trans connection. And it looks like that at least in this structure, the phosphoserine is bound in a very different way than we expected to this calcium cluster. And if you model this now, you can actually come up with a fairly nice simulation showing you that even the single C2B domain can bridge the membranes and cross-link the membranes. Well, my time is up, and I would just like to summarize. So the canonical view of synaptotagmin function, which you still find in most reviews and which is still supported by the majority of my colleagues, is that synaptotagmin, that in the docked state, before the calcium pulse comes in, the snares are already partially assembled. They're arrested either by an inhibitor molecule called complexin, or by perhaps membrane tension, whatever. And then when synaptotagmin comes in, synaptotagmin then acts on this complex either by acting on the membrane and or by the snare complex, and this way bringing the reaction to completion. Now, what we think as an alternative scenario, which we would like to discuss, is that the situation might be slightly different, that synaptotagmin might already make contact with the membranes, but the snares don't see each other before the calcium comes in, and then the calcium causes synaptotagmin to cross-link the membranes. The snares get into interaction distance and then fire and bring fusion forward. And this is the advantage of this model. It's much simpler in some ways. And it also would reserve the entire energy for the snare complex assembly for the fusion reaction. You wouldn't have to arrest it halfway, zipping off quite a bit of energy from the reaction. At that point, I would like to thank you for attention. I would like to mention again my collaborators from my lab. Matthias Hernandez did all the cryo-EM studies on the large liposomes, and I would also like to mention in particular Gert van den Bogart, who has now left the lab to take up a faculty position in the Netherlands, and we had a lot of collaborations. I would like to mention in particular Stefan Raunzer for the cryo-EM microscopy, Peter Waller for the single particle FCS experiments, and where is he? He's not on here anymore. Markus Wahl here, he is, who helped us in the solution of the transmembrane complex crystal structure. Thank you for your attention.
Great, thanks very much. And the talk is open for discussion. Ron?
Reinhard, 2 points. First of all, is the proton electrochemical gradient across the synaptosomal membrane has nothing to do with this?
Say it again?
Does the delta mu H across the synaptosomal membrane have anything to do with membrane fusion? Is it involved at all or not?
According to, I mean, we haven't really done these experiments, but according to my understanding of what has been published, the answer is clearly no. You can basically kill the transvesicle membrane potential and you still get normal exocytosis.
Okay. The other thing is, most of these membranes, from synaptosomal membranes to right-side-out vesicles from E. coli, they're loaded with protein, right?
Yes.
And most of the transmembrane, at least the multi-spanner membrane proteins, have very irregular surfaces. And usually, what's the protein-to-lipid ratio in synaptosomes? Probably
in the synaptic glass membrane, you can't tell because I cannot purify them. The synaptic vesicle is 1.9 to 1. I mean, I showed you the model. We really spent years in trying to quantify that. So,
I mean,
so about a quarter of the synaptic vesicle membrane's volume is taken up by transmembrane domains. It's quite significant.
Okay, but what this brings me to is most of these transmembrane proteins end up having very irregular shapes, and the helices are irregular, and therefore the lipids in the vicinity of the proteins can't be in bilayer configuration, at least not stable bilayers. And if these things are really loaded with protein, the lipids are not really in a rigid bilayer state, which must have something to do with fusion, I would think. No?
I agree. I should say, though, that we were surprised by ourselves when we routinely use, in addition to our liposomes, we use native membranes. So we use synaptic vesicles and we use chromaffin granules, which I've never seen any detergent. So these are membranes which we simply isolate from these particular tissues. And to our surprise, the snare, at least as far as the snare part is concerned, the reactions look very similar to the liposomes. So what I think is that the snares are basically, they are natively unfolded, they stick out there, and they are this enormous energy release. Once a zipper, they shift everything aside. I mean, that's my notion of this. Otherwise, I cannot explain this surprising similarity in the, both with respect to kinetics and everything else we've looked at in the fusion reaction when we use an artificial membrane and we use a native membrane for comparison.
So this model for the Synaptotagmin pulling the membranes together is really elegant. I like its simplicity. And don't you think there should be something like that, a non-calcium dependent version of this in the endosomal snare pathway? If so, what would that be?
Yeah, I think in a way you're right. I mean, the question is obviously, I mean, my model, if I want to call it this way, raises a couple of other questions. I mean, this means that the positioning of the vesicle is becoming very crucial. But it has one additional advantage, and this is that whatever pulls the vesicles closer should fire the snares. And there is a phenotype which neuroscientists have been struggling on for years, and this is a fact that hypertonic solutions can trigger the release of all docked vesicles. Whether you use sucrose or something else is a physical effect. With our model, it's very simple, because essentially you relax the membranes, you pull the vesicles, you create a drag, the vesicles get closer, the snares get into firing range, and the reaction is completed. This is very difficult to explain with the other models.
But could something like this be going on with the internal membranes?
In which membranes?
So in early endosome fusion, for example.
Well, there, by all, let's say, knowledge, the fusion is not regulated. So once basically what you have tethering factors, which form a bridge, and once these tethering factors proofreading with RAP proteins, and once, let's say, they are local rearrangements, and what these rearrangements are, nobody knows. But once the snares are getting close,
right. But you think things like the diesel complex,
yeah, exactly.
tethers are bringing things as close as
right.
the synaptotagmin can.
And well, this you might call it a synaptotagmin-like reaction, because there must be something which brings the membranes together. You're right.
Reinhard, the question to the very attractive idea, of course, that snares just bring the membranes to small distance, and this is sufficient for fusion, if I understand correctly.
Yes.
This is really very attractive, nice, and this is definitely a part of the story. My question is whether this is the full story. We know, I mean, if one thinks about the mechanism by which dehydration may drive fusion, we had such ideas, but there are also experiments with artificial bilayers which are brought together by pressures, by osmotic pressure or dehydration if the experiment is done in vapor. So, and we know the first results were from Huey Huang, who got this stalk phase, and then by Tim there are experiments and so on. So as far as I understand, the distances one has to reach are less than 1 nanometer. One talks about 0.8, 0.7 nanometer, and this is a distance of really of water gap. And then indeed the hydration energy is so large that one can imagine that the fusion intermediate replaces the hydration layer, and its energy is lower than that of the pre-existing hydration layer. My question is whether snares can bring membranes to such a small distance. If one thinks about the snare complex itself, which sticks between the membranes, its thickness, as far as I remember, is about 2 nanometers at least. So how a body which sticks between the membranes and has its own thickness of a couple of nanometers can still bring the membrane without bending them, without generating curvature and so on, to lower distances in order to drive fusion. The numerical experiments by Helmut Gruppmüller are not, in my view, are not so convincing because they were performed with very small vesicles, which are so strongly bent by themselves that I easily believe that if you take such strongly curved vesicles and bring them point-wise together, even by one snare complex, they will fuse by themselves because they are fusogenic because of their bent state. So the question is how flat, more or less flat membranes can be fused by just by bringing them together to distances of about a couple of nanometers, which are not fusogenic. We know it from experiment.
I think we should continue this at the beer, but I mean, just a short answer is, my very short answer is I simply don't know. I think for me, there aren't, for instance, I think Tom Südhoff has shown, it's not yet published, that you can actually replace transmembrane with a snare with a lipid anchor, and it still is functional in an intact system. So, which means in a way, and lipid anchors, I mean, you cannot have a stiff connection to the snare bundle and the membrane anchoring part. So I think I would like to know how close they're bringing them. I think with our cryo-M, we have a tool now to unravel this by using mutations and see what we find. The other issue is obviously that you shouldn't forget about local thermal fluctuations. And the final point I can make is, I mean, this kind of fact that the transmembrane domain is a bit too short, I find very intriguing. And for instance, if you start putting on, people have put on GFP on the C-terminal side, but they need to have a spacer which is uncharged. If you put on the charge directly, you really severely impair snare function. So my idea is that they might, in the zip ring, they might actually be pulled out a little bit. And this way might drag out, head groups, creating kind of local hydrophobic defect, add thermal fluctuations to this, and you might have a kind of fluctuation energy levels which might easily overcome the energy barrier. But I mean, it's a hand-waving explanation, I understand that.
Further question?
Very nice talk. And I'm curious, has anyone tried to measure the forces that can drive dissociation between T and V snares? The reason I'm asking this question is you showed, at least in vitro, one snare complex is enough to, drive membrane fusion, at least, in artificial cases in vitro. So obviously that has something to do with, I would tend to think, membrane fusion is energetically unfavorable. You need some energy, eventually enough forces to drive two membranes close to each other. And that force relates to the curvature of the membrane, as Misha alluded to. So I'm curious because, the force that drives protein unfolding is on the order of 10 to 20 piconewton, right? So my feeling is it's probably larger. You need a larger force to draw membrane together.
Yeah, I mean, these several recent studies have addressed this, not from my lab. I mean, they have looked at using the so-called surface force apparatus, and also I think with more elastic systems, snares have been pulled apart. I mean, the assembly reaction is difficult to measure, and this is, in a way, to assume, let's say, energies from pulling apart reaction is also problematic because the disassembly is associated with a large hysteresis. So there's a problem, experimental problem here. But also in terms of energies, as I said at the very beginning, we do not know the energy barrier. Let's face it. I mean, I really went through the literature, went through everything, and we don't know in KT values what the energy barrier of a normal fusion reaction is. And I think what we can do is with these kind of experiments, where we have cryo-M, we can at least, the reason why I'm excited about this is that we can actually capture intermediates. We can look at them at very high resolution, and we can actually manipulate the snares. Because this is a system we can completely control. We can manipulate the snares and look at these intermediate states. And I should say that this is also not without problems, because as everyone who deals with surfaces knows, that if you bring membranes close to each other at some point, the interaction becomes irreversible because you get hydrophobic interactions, even if they don't fuse. Yeah? So you get hydrophobic contacts, and you cannot, for instance, if you take our vesicles and we digest away the snares, we can do this in certain ways, they still don't come apart anymore. So this kind of tight opposition at some point becomes irreversible. So these are other issues one needs to address, and I don't really have a handle on so far.
So I add a very short question. In your full-length complex, snare complex, you pointed out the linker region is helical.
Yes.
And you always show in your figures the linker region quite bent. how sure are you that it's really bent, or can you just tilt the orientation of the helix in the phospholipid bilayer?
Yeah, I showed this original picture from the 98 paper. This is certainly, I mean, I wouldn't defend that model, yeah? Because whether it's a contiguous helix, even in the before assembly, I would doubt very much. In fact, there is work done, for instance, by Lucas Tam lab and other labs, Jose Rizzo lab, who looked at NMR, and there seemed to be helical propensity in this linker region, which is not contiguous. So you see the transmembrane domain in the free protein, then you see a small helical domain, which actually can, with a, let's say, with a short non-helical interruption, can actually interact with the membrane. There have been EPR studies being done showing that you have, in the free synaptobrevin, the linker is partially immersed in the membrane, even though there's, again, this is not fully accepted by everyone, whether this looks like this. But there is clearly, during the zippering reaction, at which point the linker becomes fully helical is something we don't know, probably only very late.
Okay. So you have heard that there is the opportunity for further discussions at the bar. Ah, there is another question. I don't want to cut you out.
Is there any protein that inhibits the fusion between the vesicle and the membrane?
Say it again, I didn't quite catch your question.
Is there any protein that inhibits the fusion between the vesicle and the membrane?
Well, big debate about that. So currently, the candidate protein which is supposed to block the full zippering is a protein called complexin. Complexin is a small molecule which binds to the outer surface of the Snare complex, but it has also been, there's some evidence that it might actually also interfere with the final stage of the zippering. Another idea which was brought forward that Synaptotagmin by itself might block the full zippering of the Snare complex, but at least in our experiments we have never ever been able to see any inhibitory effect on zippering by Synaptotagmin, whatever experimental system we have looked at. But it's a debated issue, I should say.
Okay, so the final question.
Thank you. Professor Jahn, I want to ask a question. If it's not two liposomes can fuse together, if the membrane, one membrane is the planar membrane, you can fix the membrane in maybe the plate. Maybe something you can do to measure the force of the liposome fusion with the planar membrane, whether it is workable.
Yeah, I mean, we are involved in various collaborations involving planar membranes, and planar membranes are not so easy because either you have supported bilayers, and then you have always a contact between the membrane and the support, which affects the membrane, particularly when you have massive movement of large, areas of lipid. And there's also, we have a collaboration with a group that forms membranes over small pores. So you have essentially porous material, you form membranes on top of it. You can look at the fusion of the pores, but it turns out that this membrane is under tension, like also black lipid membranes are there under lateral tension. So it's not a trivial problem, experimentally speaking.
Okay, thank you.
Okay, so I'll thank the speakers again for their contribution, and all to you to the contribution to the discussion.